So I first want to thank Logan for inviting me to give a talk here. It's my pleasure to be here and giving an overview of how to use nanophotonic detection for drug binding to membrane proteins and use ultra-sensitive methods, and that's something as a protein biochemist we are always looking for. So Yes, changing to the next. Okay. So first I'll give a brief overview of my research group. So I've been a faculty here since last October, and I have 4 graduate students working in my lab. As you can see, they're from different departments as my lab requires different expertise. The person who will be working on the nanoplasmonics in my lab is Snaitha from the bioengineering department and Eric Magneta, an undergraduate. So we are in the vet school. Vet school is a little bit away from the main campus. I'm sure none of you have seen it. It's a little separate from the campus, maybe 15 minutes away. It's quiet, and primarily there are the veterinary students. So this is my research group. We are a biochemistry lab, and you can see a lot of typical biochemistry instruments are here. We express proteins in E. coli, insect cells, and human cells, and then we take those proteins out. We put them in the structures called nanodiscs, which I'm going to describe today what those are. And then we study 2 things in the lab. First is we try to study their biochemistry of how this protein function. So we are those people who study in details about proteins mechanism. And the other things my lab is interested in is combining ultra-sensitive nanodetection tools for looking at drug binding to the membrane proteins. And I will explain why we do that and why we are interested. And there is a huge need of more ultra-sensitive devices to be coupled with drug binding to membrane proteins, because that's sort of a need right now, both from the biochemist point of view, from a doctor's point of view, and overall. So I'll actually first start with the acknowledgement slide. This work, which I'm going to present today, has been done in collaboration with Northwestern University and Dr. Rick Van Dyne's lab, and Professor George Schatz, who has been our theoretical person who made us understand what we are observing, the new phenomena we are seeing, and Dr. Jing Zhao just recently started her faculty position. And I also want to thank the Sligar lab, where I did my postdoctoral research, and in particular Dr. Denisov Brinkova and Dr. Mark McLean. And I would like to thank for support the Startup Research Funds and the NSF Nanoscale Science and Engineering Center at Northwestern, where a lot of collaborative research started, and this work was part of this collaborative research. So as I said, the main research directions in my lab are understanding the membrane protein mechanism, how does protein function, why do they function the way they do, what are the important residues or peptide amino acid residues in the protein, why are they important. The other part of my lab works on surface-based detection, and then we are also interested in therapeutic intervention. That is, suppose a person has an Alzheimer's disease, why do they have it, what proteins are involved. So to understand the basic understanding of why, what happens, so that we can have new development of a new therapeutic target. So right now we are looking at 3 targets in my lab. One is the CYP2J2 epoxygenase. That's actually when the cancer metastasizes, this protein is 7 times overexpressed, and we are trying to study why this protein it leads to the increase in cancer metastasis. Then we're also working with COX. I'm sure all of you have taken aspirin and ibuprofen at some time. But this drug molecule interacts with this COX protein, and that's a very key protein for inflammatory response. And we also study thromboxane synthase, which is CYP5A1, which is involved is a heart attack. It's overexpressed when you're having a heart attack, stroke, myocardial infarction, etc. So today I'll talk more about the surface-based detection methodologies. So why are we interested in membrane proteins? So we always hear about all these diseases, like take for instance breast cancer, Alzheimer's disease, cystic fibrosis, heart diseases. But what we don't appreciate is when we talk, we say proteins are involved in these diseases. But actually these are just not protein by itself, but these are membrane proteins which are involved in these diseases. And a single mutation in these proteins, like in cystic fibrosis, it's just one mutation which causes this disease. And so you have to appreciate that it is a very important to study membrane proteins, and they are difficult to study outside the membrane environment and outside the cell environment. But if you don't study the biochemistry of this protein, you cannot develop drugs, and you cannot use nanodetection tools to, for diagnostics. So this shows the targets of the current drug therapy, and you can see that more than 50% of these major targets are membrane proteins. But in our body, we just have 20 to 30% proteins are membrane proteins. So that means that they are very important targets, and naturally the question rises, why? Why are they such important targets? What do these membrane proteins do? So this is a schematic view of a cell membrane. So the yellow here shows this is the lipid bilayer, and these proteins are these membrane proteins are embedded. So they are like gatekeepers of the cell. And molecules, drugs, anything from outside the cell wants to go in, has to talk to these gatekeepers. So now that's why they are challenging to study, because if you notice that these lipid bilayers are actually hydrophobic in nature, while these membrane proteins have to be partially hydrophobic and partially hydrophilic to function. So they have to be hydrophilic because they're interacting with molecules outside the cell membrane. They have to be hydrophilic inside because they are now shuttling this molecule from outside to the inside and interacting with some proteins inside the cell. But they have to be sitting in the lipid bilayer to give them stability and to form this gatekeeper function, and therefore they are hydrophobic too. So what happens if you pull out a molecule which is partially hydrophobic and hydrophilic? Now you cannot use completely hydrophobic solvents, you cannot use completely hydrophilic solvents. So you need somewhere in between, and that's why the studies have been challenging, because people were trying to find what can we use in between which can solubilize these membrane proteins. So if you look into the cell structure, we have lots of membranes. We have the cell membrane, we have nucleolar membranes, so many ER membranes, so many different membranes. And just as I said, this is one of the proteins. It's a membrane protein. You can see it's the hydrophobic domains of the membrane protein. Now, if you're trying to pull them out of the membrane, what you end up is something looking like a scrambled egg. So it's quite a mess. So what are the different methodologies to solubilize this protein? Because if we cannot solubilize this protein, we cannot study their mechanism, we cannot put them on surfaces. So both these, so stability on solution and stability on surfaces depends on how we are solubilizing these proteins outside the membrane environment. So this is the different solubilization methods, and people have commonly used detergents. So we all use detergents to clean our clothes, and what the detergent does is it interacts with the grease. It also has a hydrophobic and a hydrophilic interaction, and that's what the detergent is actually shielding the grease part of the protein. But the problem with detergents, it's unnatural. Some detergents looks like drug molecules actually, and they actually go and bind to the active site of the protein. They denature the protein. So there's a loss of function. And if you take out a membrane protein in detergents, it can be unstable over a day. Like you start an experiment in the morning, by the time it's 8 o'clock, your experimental system itself is different. So you don't want to, so the data people get on membrane protein can be varied depending on the lab, depending on the graduate student who did the experiment, and you don't want that variability if you really are studying something, right? So the other method people use are liposomes. So liposomes are lipid bilayers. The green here shows the lipid bilayers. It is like the native lipid bilayer, looks a little bit like a cell. But the problem is the liposomes are, in membrane proteins, there are functional domains both sides of the protein. So in liposome, one side gets inside. There could be 3 or 4. There's a heterogeneity involved in the liposomes. And they're also prone to aggregation. So a newer method developed by the Sliger lab is the Nanodisc method. So nanodiscs are, so in our body, the thought was that in our body, high density lipoproteins carries cholesterol. We are always worried about our high density lipoproteins. So instead of a cholesterol, which is a hydrophobic molecule, let's replace that with maybe a hydrophobic protein. And actually you can replace that with a hydrophobic drug, anything. And then this HDL particles becomes a carrier of not only cholesterol, not only a protein, but could be of any other hydrophobic molecule. And that's what this does. So nanodiscs can be used for solubilizing the membrane protein. So now this is a lipid bilayer. It gets sort of shielded. There is a membrane scaffold protein which actually sort of controls the shape of the, controls actually the oligomerization state. And now you have both sides accessible and it's soluble. It is so good that you can actually treat it like a soluble protein. So now you have a protein which is soluble, and now you can study, and there's less heterogeneity. And any lab who started working on this has stayed on with this, stayed on using nanodisc for membrane protein study. So what are the components of this nanodisc? And this can be a tool you can use for many other things. That's why I think it's good to understand about this. So one is the membrane scaffold protein. So you can imagine this is like a sushi, just think of it like a teppanyaki. So you can have the membrane scaffold protein, which is around the nanodisc. You have the phospholipids, which sit inside the of this membrane scaffold protein, and you can actually put the membrane protein inside this lipid bilayer. These are quite uniform. This is the AFM image, and this is a size exclusion chromatography, and you count the number of lipids in the nanodisc, and it's quite uniform across the whole peak. So there have been a lot of different membrane proteins which have been put into nanodisc, and quite successfully. And you can learn a lot about these proteins by putting them into nanodisc. Suppose you ask this question that are this is a TAR receptor. So you want to know if one TAR receptor can signal or 3 TAR receptors are required for signaling. So if you put it in nanodisc, the 3 signals and the 1 won't signal, and there's actually a kinase activity involved with that. Then this is a protein translocon, so that is, it shuttles protein from outside the cell to inside the cell. And again, this is this can show that kind of a function like in solution in vitro. What my lab studies is we are more interested in cytochrome P450s. These are drug metabolizing enzymes, and I'm going to talk more about it. I'm interested that drugs should bind to it, but another point of these proteins are these are expensive to purify, because when you it will take a grad student almost 2 weeks to get enough proteins for one experiment. So these are expensive. We want to have ultra-sensitive detection methodologies. We want to use less and less protein and get more and more information out of what is happening, how is it functioning. So that's why our interest is, why we want to couple it with the nanodiagnostic tools. So if I summarize the section, we can say that the membrane proteins, they play a very important role in health and diseases, and there is a strong need for the ultra-sensitive devices for detection of the drug binding to the membrane protein. And then we learned a little bit about the nanodisc, that it can stabilize the membrane protein, and both on surfaces and in solution. We always knew about solution, but my work was more involved in how to see that the nanodiscs are stable on surfaces too. And then we can actually couple membrane protein and nanodiscs with different ultra-sensitive devices, which I'm going to show in the next slide. So these are part of what we were working on, and we showed that actually you can, if you take just a detergent solubilized membrane protein and couple it to an ultrasensitive device, you will get no information, because that protein will actually collapse. But you can use a lipid bilayer, like an artificial lipid bilayer, and lay it on the surfaces, and that would be more stable. And with the nanodisc, the advantage is you need not actually, the lipid bilayer even need not interact with the surface, and you can use the tag on the scaffold proteins to put those membrane proteins on the surfaces. So the different methodologies where the membrane proteins in nanodiscs were combined with the different ultrasensitive detection methods are surface plasma resonance. So surface plasma resonance is a very key tool which protein biochemists use a lot. They use it for understanding protein-protein interaction, protein small molecule. Actually, it cannot be very small molecule. It has to be around above a certain size. So SPR is a very important method, but again, they could not study this for membrane proteins in detergents, and the nanodisc was very good for studying them. Then we also used cantilever-based detection methods. So sometimes the membrane protein targets are not optically active. They don't have any chromophore. So you need a technique which does not depend on optical signal, and therefore that was the thought why we wanted to couple it with a cantilever-based detection methods. We also used self-assembled monolayer MALDI, which is you put the protein on the surface and then you do a MALDI on that to figure out what protein got activated, what bound to it. And that's a very important tool. And then we also coupled it with microfluidics. But today I'm going to talk primarily about the using of localized surface plasma resonance and surface enhanced Raman spectroscopy on drug binding to the membrane protein in the nanodisc. So what is localized surface plasma resonance? I'm sure all of you know about it. And localized surface plasma resonance is a very attractive detection method, and it is because you can use small nanoparticles. There's an ease of fabrication. You can take it to the patient's bedside, and it's more simple compared to many other devices which might require more complicated detection methods. So bulk gold typically have a nice yellow shine, but these nanoparticles embedded in this Lycurgus cup shows that the scattering spectrum is different, and the absorption spectra is different. And these nanoparticles, you can vary the sizes and you'll get different colors. So the type of nanoparticle we use in our study is fabricated using Nanosphere Lithography, developed by Hultin and Van Dyne's lab. So how this fabrication works is you take a clean glass substrate and you use polymeric nanospheres of different diameters, and then you can dry those substrate on the polymeric nanospheres on the glass substrate and deposit silver. And when you sonicate it, you remove these polymeric spheres, and what's left behind are these nanotriangles. So if you do the AFM image of the polymeric spheres, you see they're quite uniform. And if you do the AFM, so these are like the nanotriangles, and you can vary the size and the shape of the nanotriangles, and that makes their spectra vary as well. So the detection method here is very simple. This is the Nanosphere LSPR sensing. So you can use a simple UV-vis spectrophotometer. This is the nanoparticle substrate. You can put it in the UV-vis spectrophotometer. You can also measure the scattering using a dark field spectroscopy. And we have, initially we have been doing this, and now we were moving to more of using dark field spectroscopy. So the nanotriangles are, I said, that the variation of the shape and the size can change the LSPR of the nanoparticles. So if you see that if this is a little roundish in shape, more triangle, more sharp, then there's a change in the colors, and the spectra of these vary. And this is very important for our studies because we are trying to study that we have a colored protein and a colored nanoparticle, and we are trying to study their plasmon resonance coupling. So we did fabricate different nanoparticles which have different absorption spectras. So what is the LSPR wavelength shift spectroscopy? So if you have a glass and you have the silver nanoparticle, consider this as a non-resonance adsorbate, then the shift will is directly proportional to the refractive index change. But what happens if this non-resonant molecule is now replaced with more of a resonant molecule? So when you have a resonant molecule, the story is a little different. So now you consider this as a resonant molecule and you measure the shifts. And I'll show you how the shift will depend on the spectra of the resonant molecule, and depends also on the nanoparticle initial plasmon resonance. So we are measuring the LSPR shift, and we studied with different resonant molecules with Mg, it's a pyrazine, and the iron bipyridyls and rhodamine 6G. And we are going to primarily measure the LSPR shifts. So this is one of the molecules, which is the magnesium porphyroxenato. So this has a spectra which looks like this. It has a maxima at 597. So this is the solution spectra of this molecule. Now what happens is, suppose we immobilize these particles on the nanoparticle surface, and what we plot here is the wavelength. So this is the wavelength maxima of the nanoparticles. So these black dots are basically different nanoparticles, and what we plot on the y-axis is the LSPR shift, and the green here is the spectra of the molecule itself. So what we notice is like average shift of these nanoparticles as a delta lambda on binding these resonant molecule is around 20 nanometer, and the maximum shift happens when it's a little positive, little red to the molecular resonance of the molecule, and then the minimum LSPR shift happens when the molecular and the plasmon resonance are coincident, and the amplified LSPR shift is almost 60 nanometer, which is not expected if this molecule was not a colored substrate. So if you had a non-adsorbent substrate, it would be approximately 20 nanometer average. So to make the point again, so you see that here the average shift is 20 nanometers. So this is the delta lambda, and if you have it overlap a little bit red, it's not red, it's right on, then it's almost 2 nanometers, and then the shift changes if you have a 60 nanometer shift when it's a little red. So that means there is a plasmon coupling happening between the colored adsorbate and the nanoparticles. So how general is this phenomena, and can we use it for something more biological? So we also checked it first on iron bipyridyl, and we saw that same kind of phenomena can be observed for bipyridyl. And here also it's even more redshifted. You get more changes, and it dips down when it's at the same wavelength, and it's average around 10 nanometer shift. So we see that a large redshift happens when the nanoparticle LSPR is located at a slightly longer wavelength than that of the adsorbate molecular resonance. And the resonant LSPR response opens up new possibilities for detecting binding of low molecular weight analyte to a colored protein. So suppose this molecular weight is the molecular weight of this drug is very small. You cannot use SPR because the refractive index change would be very less. But what you can use is you can use the LSPR because there is an enhancement when the spectra changes. So that's what you're taking advantage of. I'm going to talk more about it. So that's what we are interested in. We are interested in detecting the drug binding of a low molecular weight, which are like some small molecules like imidazole, camphor, to a colored protein molecule receptor. So what would be the best colored protein we were looking for, which would be, and to apply it. And one of the proteins we all have, actually, or not only we, everybody has. So this is the Cytochrome P450 superfamily. And if you look into this you can see that bacteria, eukaryotes, archaeas, everybody has a P450. So what are these P450s? So if you have talked to an organic chemist, they'll complain that doing the CH activation is a very difficult work. Like they are all working on CH functionalization. But these P450s are our body's organic chemist. They convert CH to COH, and they do it all the time. So they basically oxidize CH bonds. And this reaction is so key for different organisms that all the organisms have this protein. Although it's a class of protein, these proteins are quite different from each other and might share only 20 to 30% sequence homology. But they are similar to look at. So it's very interesting, and there's some people who study how proteins look like. So these proteins are typically always triangular in shape, and they think that the substrate access channel is always from this side. Maybe that's why they have a triangular shape. They have a heme binding site, and that's what gives them this beautiful red wine color. So it's a pleasure to work with these proteins because they are always red wine in color. And when my students work with the non-colored proteins, they are like, it's difficult to see them because so now you're running gels, you can always see these proteins, the nice red color. And that red color comes from this iron heme protein, which binds to the protein active site. So that's how the active site looks like. There's the iron in the middle. So here I show the spectra of CYP101. The nice red color comes from a Sorey band, which is the green here, at around 417 nanometer. It will vary from 415 to 417 depending on the protein, quite standard. There are also Q bands, resonance. And then what happens is if you bind certain type of molecules, which are called the type 2 molecules, and they are inhibitory in the nature because they actually bind directly to the iron center here. Those molecules produce a red shift. So here the red, you can see the red is actually from the green to red shift. And then there are other class of molecules which these proteins metabolize, and these are called the substrates. They always produce a blue shift, so that's green to the blue shift. And so that means this is a great test study we can do, that we can combine it with the LSPR sensing, and we can see if really this resonance coupling is happening, and if we can actually detect the small molecules. So imidazole and camphor is very difficult to be, you cannot detect the binding with other methods. And in the solution spectra, you're using a lot of protein. It's almost a 1 micromolar solution, excuse me. So that's what we did. So we took the silver nanotriangles. We modified the surface with 11 mercapto undecanoic acid, and we varied the ratio of 11 mercapto undecanoic acid and kept some without any acid group at the end so that we can control the number of proteins on the surface. And then we EDC coupled the protein. EDC is not the best way of coupling proteins on surfaces because it tends to denature. But here this protein has its internal check that if it will not bind the substrate if it's not folded, and which is not always true for every protein. So it has an internal check. So we went ahead and tried the EDC coupling, and this, as this is a localized surface plasma resonance, it's just decay length is 50 to 100 nanometers, so it has to be very close to the surface. And then we bound drug molecules. Here is basically a test compound because this is a test protein, is the camphor and the imidazole. And the protein we were studying is cytochrome P450 cam. So we see a very similar phenomena. So this is our step 1, where we add the protein to the surface of the nanoparticle, and we see that this is the protein spectra, and these are the nanoparticle shifts, and it is a little difficult to fabricate nanoparticles this blue because they start to fuse together. But so after several attempts, we were able to fabricate few particles which were these blue. They were absorbing around 400, 410 nanometer. So you can see that when it overlaps, there is a dip, and when there's a huge enhancement, almost 60 nanometer when this is slightly red, and then again the average comes around 20. We also see another plasmon resonance effect here. There's a coupling here, this huge shift, and you would think, why is this huge shift coming? This is actually coming from the Q band. It took us few, but we were unable to explain why the shifts are, the extent of the shifts are similar to that of the Sorey. So that's sort of we are still working on it. So we saw that there's a 66 nanometer shift, which is quite a lot of shift on the nanoparticle resonance when it's a lightly red. I remember this particle being around 420 nanometers, slightly red. 417 is the spectra of the Sorey of the protein. And so this was what we were, okay, so that's for the first step, and we saw that we kind of see the same phenomena as we saw for the different dye molecules we studied before. Now, what do we see when we have the drug molecule? So now when the drug molecule binds, in solution we see a positive shift for 101 with imidazole, and with camphor we see a blue shift. And interestingly, that's what we see when we do these studies in on the surface. And this was actually the first time anybody could see a blue shift. So we questioned our experiment many times, because you always have a positive refractive index change when something binds to the molecule, and you always see typically a red shift. But we saw a blue shift when camphor was binding, and that could be explained because we see a blue shift, the net blue shift, when it binds to the molecule in solution. But that means the surface is like reflecting what's happening in the protein. Like the protein is binding, changing its spectra, and the LSPR, the nanoparticle, is acting as a detector. So the camphor, you can see that this is a blue shift from A to B, and for imidazole we see a positive shift. So this shows that it's the same shifts we see in solution versus the surface, and we see the red shifts at solution versus surface. So can we explain this? Why do we see a blue shift or a red shift? We can qualitatively explain it. We cannot still explain the extent of the shifts we see, like 66 nanometers is a huge shift to explain. But we can qualitatively use, understand what might be happening. So this delta lambda max is actually dependent on the refractive index change, and the refractive index change depends of the non-adsorbate and the resonance adsorbate. And if you plot, this is a sort of theory, that if you plot the change in the refractive index for these molecule binding imidazole and camphor, then you do see the predicted LSPR shifts are positive and negative, and that's what we are seeing in the real experimental data. But what we could not explain is the extent of the shift. We are seeing more shifts than the theory predicted, and more detailed theory is required to explain that. So if I summarize this section, we see that for binding a non-resonant protein, there's always a red shift in the LSPR. But if we have a resonant protein, there is an observed wavelength-dependent shift in the LSPR. And so the LSPR is sensitive to the electronic structural changes of the adsorbed species, like whatever is, like if you studied this with a dye molecule, you will never know that it can actually translate, or it could be sensitive to what's happening in the protein. But because the protein spectra is changing, the LSPR is acting as a reporter group. So the thoughts on this is that the extreme sensitivity of the LSPR wavelength to the adsorbate electronic transition makes it possible to use this technique to detect low molecular weight adsorbates. And there are very few methods which actually detect low molecular weight adsorbates, but there's always a need for that. So now I'm going to talk about applying this to a real life problem, and something which we take every day, drugs, and something which older patients, HIV patients, take more, and it could be a problem. So what we are interested in is screening drug molecules to binding to human P450. So now we're going notch up from the bacterial P450 we are studying, where we understood the phenomena, we know this happens. Now can we apply to a real life problem? So what is drug-drug interactions? I'll let you read this. And so if you think grapefruit juice is harmless, think again before you're taking a drug with it. So what grapefruit juice will do is it will inhibit some of the enzymes in your liver, and it will increase the effective drug concentration. And so there was a study where people were trying to use grapefruit juice to mask the drug taste. But they were seeing that there's a weird result, that all the patients who are who are taking the grapefruit juice, the drug levels are higher in them compared to the other, and they didn't want to report. They thought they'd gone wrong. What could grapefruit juice do wrong, right? So you always have to follow the proper dosage. If you see the doctor gives you proper dosage, they also try to give you like details of what shouldn't be mixed with what. If you're taking an antidepressant, you shouldn't take that, and all this mixture. You might think, why is this important? So patients, like elderly patients and HIV patients, people on chemotherapy, like who take multiple drugs, what happens is your liver has many P450s. I'm going to show you a quick slide on how many P450s you have. So what happens is one drug will inhibit those proteins, and the other drug will increase the activity of the protein. And you want to know beforehand what that drug is doing. So you want to screen for multiple drugs. And in a way, it's personalized medicine too, that you want to screen multiple drugs interacting with these proteins and know really what they do in a high throughput fashion. And that's what the pharmaceutical industry does. They screen for drugs. So this is what the drug-drug interaction is. And it can be quite toxic for many people. You might hear that this person had this drug X and they fainted and been in coma, and then they will say, why is that? Because they might have a different P450s. So P450s also differ by ethnicity. We might have different P450s. Different ethnic groups have different P450s. Depends on the individual. Some individuals have different P450s. Others have different polymorphisms. There's a huge amount of polymorphism in this drug metabolizing enzymes. And these P450s are also very promiscuous P450s because these have big active site. They have to metabolize xenobiotics and multiple drugs. So they don't know which is going to be thrown to them. So they have to be, so you have heard of one enzyme, one substrate hypothesis, that an enzyme binds one substrate. But these are exception. One enzyme can bind 3 drugs. So one protein can bind 3 drugs at different parts, metabolize all 3 of them with a bad efficiency, but at least they are metabolized. So these are very special proteins, but primarily what they are used for is drug metabolism, which I was talking about, and that's what this will have an application for. But they also make steroids. So they synthesize different steroids, and they also are involved in fatty acid and prostaglandin metabolism, steroid metabolism, and vitamin A metabolism. So primarily metabolism, synthesizing compounds for you, the testosterone, the estrogen, all these hormones we have are synthesized by these molecules. So they're extremely key. So this is these are the chart of the human cytochrome P450. We actually have less. Plants have 2,000 P450s. We actually have just 57 to 63 P450s because we can run away from danger. So things which cannot run away from danger have more P450s to metabolize the drugs. So I'm going to talk about the 3A4, which is primarily expressed in the liver and is involved in 50% of the drug metabolism. And my lab works on 5A1, 2J2, and other few other P450s. So what do these P450s do in the human? They are involved in hepatic drug metabolism. So they can metabolize all these things we eat, and they actually keep your body free from like toxicity, which you will have if you get rid of this, knock out this P450. And they're also involved with steroid biosynthesis, and sometimes it can be bad, like in breast cancer, this aromatase protein is overexpressed, which synthesizes too much estrogen, more than the body could handle. So it's the cytochrome P450-3A4, to which we are going to study drug binding to this protein using the localized surface plasma resonance. You can see this is a very important protein. 36% to 50% of the drugs are metabolized by this protein. And this is a membrane-bound protein, and that's what increases its complexity. So CYP3A4, that's why we put this in Nanodiscs, which I introduced before. So CYP3A4 can be incorporated to Nanodisc with a controlled environment. This is just exactly like a sushi, like this protein sits inside the lipid bilayer. There's a membrane scaffold protein. It's a simpler system to study, and there is a enhanced stability of this in surfaces and in solution. So how do we assemble these proteins in a nanodisc? So we have the phospholipids, we have the protein in a detergent, we have the membrane scaffold protein. So biobeads or detergent are something which removes detergent will process this self-assembly. So this is a self-assembly process, so there is like no external covalent modification, and this the way it forms easily, they can also break down if you give it detergent, so they'll just open up and release the proteins, and then you separate them on different columns and you have a population of this nanodiscs. So now how to couple that to the LSPR sensing mode? So this the first we tried was, we'll always try something simple, right? We just took the protein in detergent, put them on the surface, and we saw no change. There was no detection at all. So then we were skeptical that this is just a 50 nanometer and this is quite a big amount of stuff you're putting on the nanoparticle surface. Will we be able to see shifts and what will be the coverage? So we calculate the coverage is around 100 nanodisc molecule per nanoparticle. And we were again coupled it with the EDC coupling, and we just hoped that this was the membrane scaffold protein which was interacting. We tried more interesting coupling chemistries, but they were all putting the protein, taking the protein away from the surface, and it's a quick decay because it's a localized surface plasma resonance. So this protein again can bind type theoretical theory, right?
So the scaffolding membrane, can you produce it or is it available?
So the scaffolding protein, we produce it, we have a patent on it, but it is like an ApoA1, which is what the high density lipoproteins in our body is made up of. And you can assemble those with ApoA1 as well, but the ApoA1 won't give such a uniform disk which is done because we have optimized the scaffolding protein to make it very uniform.
Is it compatible with some artificial polymer? Like, can you put them together and they can stay together?
Yes, that is actually one part which I am interested in is instead of the scaffolding protein to use polymers, because that will improve a lot of things. It will make it more feasible to use NMR and other methods for detection. But there is I would say a competing technology called amphipols. They use polymers and they build bilayers. So that's sort of competing, but it's not exactly like the nanodisc.
Okay, there's no more question. Let's